Welcome back to MLT 120. Today we're going to be discussing abnormal red blood cells or erythrocytes. So instead of following along with the PowerPoint today, we're going to be following along in your atlas. So you'll need to turn to page 90 and we're going to be going all the way to page 106 in your atlas. This will cover chapters 10 and 11 in your atlas. So let's look at page 90. We're going to be talking about variations in color and size in chapter 10 in the atlas. But there's a term that you need to be aware of. It's called anisocytosis. This is when red blood cells have a variation in size. So here's our term, anisocytosis. So let's look at some examples of anisocytosis in our textbook. Again, we're looking at red blood cells. So this first image, image A, would be an example of a macrocytic or a small red blood cell. So the arrows are pointing to red blood cells that are on the smaller size. And you may see this cell here in the center of the image. That is a white blood cell, and they place that there for a reference because this is a white blood cell and the darker center is a nucleus, and we're going to be using that nucleus as a reference for our red blood cell size. This is actually a lymphocyte, and we can always use the nucleus of our lymphocytes as a gauge or a guide to about what size our normal red blood cells should be. So if we can see that this red blood cell is smaller than the nucleus of this lymphocyte, then we know that this red blood cell is macrocytic or smaller. Okay, let's hop over to figure B. These are normocytic or normocytes, meaning that these are normal sized red blood cells. So we can kind of compare the normal to the small. So again, we've got that lymphocyte there for reference, and we're looking at just the nucleus, the dark part, as our reference. So the nucleus is roughly the same size as the red blood cell. So we know this is a normocyte or a normal sized red blood cell. And then finally down here at the bottom, we have our macrocytic or macrocytes, which are red blood cells that are larger in size than they should be. Again, we have our lymphocyte here for reference. Again, we're looking at the nucleus, the dark part. You can see that this red blood cell is obviously bigger than the nucleus of the lymphocyte, so we know that we can categorize this as a macrocytic red blood cell. Also listed, we have some different conditions that are associated with our microcytic red blood cells, and then down at the bottom we have some conditions that are associated with our macrocytic red blood cells, and you can kind of look over those on your own. Moving on to page 91, we're still talking about anisocytosis, but in figure A, we've got what we call heterogeneous populations of erythrocytes, meaning we have different sizes of erythrocytes in the same field of view. We have some microcytes, and we have maybe some normal size cells, and maybe even some macrocytes. So let's point some out. This probably looks like a microcytic cell. This one may be normal sized, and this one looks like a macrocytic cell. In figure B, when you have two distinct populations or two different sizes of red blood cells in the same field of view, it's known as a dimorphic population. So in this one, I think we have some small red blood cells and then some normal sized red blood cells. We have our lymphocyte here for reference. Again, we're going to be looking at the nucleus. So we can already tell that we have some microcytic red blood cells, some red blood cells that are smaller. So that accounts for one population. And then this population here looks pretty close to the size of our nucleus, so I would say those are normocytic or normocytes. And I would even say this guy up here this is considered a dimorphic 2 population, but this one right here can almost be pushing to be macrocytic. Again, below each figure, you have some disease conditions that are associated with the different figures or red blood cell sizes. So look over those on your own, and we'll move on to the next page. On page 92 in your atlas, we're going to start talking about different colors or variations of color of your erythrocytes. Again, we have our lymphocyte just here for reference, but let's go ahead and look at our red blood cells. We can go ahead and tell that our central pallor in these red blood cells look washed out. So you don't have as dark of a pink reddish color as you normally would have in a normal red blood cell. So these are known to be hypochromic because they do not have as much color. These guys over here, you have some target cells, plus you have some of those same looking cells that are kind of washed out. Their central pallor is not as prominent, and they just kind of have that pale appearance. This would be a dichromic population because you have variations in color. Let's look at the bottom of page 92 at figures C and D, but let's go ahead and look at figure D. These are your normal red blood cells. These are the color that your red blood cells should be. So these are normochromic erythrocytes. They have that nice pinkish red color. You can see the central pallor a little bit, but it doesn't look washed out like those hyperchromic ones. You can also have red blood cells that are known as polys or have polychromasia. They kind of have a bluish purple color, and that's what these arrows are pointing to here. And that's because of the retained RNA that's inside of the red blood cells. Remember, our polychromatic erythrocytes will appear this bluish purple color, and they can be released out into the peripheral blood. So if you do see any of these on the smear, they will appear this way. Now we're moving on to chapter 11 in our atlas, where we're going to talk about variations in shape and the distribution of our erythrocytes. Now everyone should be on page 94 in the atlas, and the first term that we need to be aware of is poikilocytosis. This is in general a term that means an abnormality in the shape of our red blood cells. So just abnormality in shape. So we were talking about size before with an esophagocytosis. So poikilocytosis is talking about the abnormality of shape. Okay. The first abnormal shaped cell that we're going to talk about is the acanthocyte. The more common term for the acanthocyte is known as the spur cell, so you can use those kind of interchangeably. The acanthocyte is this type cell here, and the reason that it's referred to as the spur cell is because it has these little spikes that come off of it. Both these are, figure A and figure B, are images of acanthocytes or spur cells. Figure C on page 194 is also showing spur cells. This is still considered a spur cell. The spurs are just not quite as sharp. Beware of some of the disease conditions that are associated with your acanthocytes or your spur cells that are listed here. Next up are our schistocytes. These are our blood cell fragments, and there's a few different types of blood cell fragments in this category. There's just the plain old, what we refer to as a schistocyte, which is obviously just a fragment or part of a red blood cell. And figure A and figure B are pointing to some examples of schistocytes. Again, these are just fragments of our red blood cells. But down in figure C and D, these are also fall in that schistocyte or blood fragment category. These are bite cells. This is a red blood cell where this little bite has been taken out. Also, this is an example of a bite cell. And this is what we refer to as a blister cell. it looks like it has a little blister because it has this little connector piece right here. It looks like it has a little blister, just like you would have a blister on your finger. It has this little raised up area. Okay, these all fall in that schistocyte category. Something to note down here, the bite and blister cells are a result usually of your spleen pitting out Heinz bodies. Remember, your spleen has the ability to take out or even pit your red blood cells. So the spleen has taken out a portion of this red blood cell. That's why it's appearing as a bite cell now. The next shape of red blood cells that we're going to talk about is on page 96. This is the chenocyte, or we also refer to this as the Burr cell. These look very similar to our acanthocytes or our spur cells, except these little guys just have these little tiny spikes, which we refer to as more of a burr. They don't have that really extenuated spur that the spur cells have. They're usually evenly distributed little spikes all the way around the red blood cell. Sometimes they are smooth in appearance, but they're usually more evenly distributed versus the spur cells or the acanthocytes. Again, echinocytes, Burr cells, like I said, more evenly distributed projections, and they're usually not as long and as prominent. And you have some different diseases or conditions that are associated with your Burr cells listed. The next shape to cover is our spirocyte, which is on page 97. These are our round red blood cells with no central pallor. These are usually microcytic, meaning they're smaller. So if you look at the arrows or the red blood cells the arrows are pointing to, you see that these are smaller. They look more dense or darker in color, and they're just more round, and you do not see that central pallor in these guys. And you can see some of the conditions that are associated with spirocytes. Of course, we have a hereditary spirocytosis that we already talked about with our anemias. On page 98 of the atlas, we have our target cells, also referred to as our podocytes. We can also think about these as our Bell cells, because if you look at them from the side, it would be more of a bell shape, okay, like in figure A, and the A arrow is pointing to more of the bell shape from the side view. And these guys look just like you would expect them to look as a target cell. They look like a little bullseye in the center, and they have a ring, a second ring, and a third ring to make it look just like a bullseye or a target. And here's some more examples over here. And a lot of times you'll see these in our sickle cell patients. These cells have a decreased hemoglobin content, making the cell much thinner. On page 99 is our sickle cells, or our drepanocytes. Again, we can use those terms interchangeably. These are our cells that are actually sickle in shape, and you can see examples right here in figure A and also in figure B. They're long and narrow and pointed on the ends, and these will be found in our sickle cell patients. Down in figure C, you will see what's really qualified as a schistocyte, but sometimes we get these confused between our sickle cells and our schistocytes. But make note that these resemble our sickle cells, but these are actually a schistocyte. The central area is markedly thicker, where our sickle cells are very thin and elongated. This one is not. You have the pointy ends, but this center is not thin and stretched out like a true sickle cell. So that would be in the schistocyte category. The next couple of pages are our hemoglobin C crystals and our hemoglobin SC crystals on pages 100 and 101. We've talked about these a little bit in the past. Our hemoglobin C crystals are our ones that look like the bars of gold or the Washington monuments. These are the long, thin, and they usually have a rounded edge on each end. And again, we refer to them usually as our bar of gold or Washington monument, and these are associated with our hemoglobin C patients. Also, something to note about these crystals, they are known to be intra-erythrocytic crystals, meaning they're inside the red blood cells. So just remember that, intra-erythrocytic, inside the red blood cells. So moving on to page 101, these are our hemoglobin SC crystals. Remember, these are our irregular shaped crystals that are associated with our red blood cells. These can be any shape. A lot of times they take a shape of what we refer to as a little mitten. This is a pretty good example of a little mitten. It's kind of upside down. This would be the thumb and the fingers. But again, they could be completely irregular in shape. So here's some examples on figure A, B, and C of the SC crystals. And again, it's associated with the hemoglobin SC disease. On page 102, we have examples of the elliptocyte and the ovocyte. Figure A is the elliptocyte. These are usually what we refer to as cigar-shaped red blood cells. They're kind of long and narrow. So there's some good examples on figure A. The ovocytes kind of look egg-shaped. And there's some good examples in figure B. And you can see some disease conditions associated with both of these down here on the bottom of the page. On page 103 in our atlas, it's talking about the teardrop-shaped cells, or the dacrocytes, and these look just how you would expect them to look, like a teardrop or a drop of water. They have one pointy edge and a nice round end on the other. And figure A and B is giving you both examples of our teardrop cells. They're associated with some of our thalassemias and anemias. On page 104, we have our stomatocyte. This red blood cell is said to look like a little mouth, or a little coffee bean. Both have been made as comparisons, but it's similar, like I said, to a little mouth. Sometimes it looks like they're smiling at you because that central pallor is kind of smooshed or bent to make this little smiley face or the little coffee bean shape. These are associated a lot of times with liver disease or alcoholism, or even a hereditary stomatocytosis. The last couple of pages that we're going to talk about in our atlas today are talking about distribution, and rouleau is something we've already talked about a little bit in relation to ESR testing, but rouleau is just when we have our red blood cells stacked together, and it's usually referred to as a stack of coins, and you can see examples in figure A and figure B of how red blood cells can stack together. Also, when you get to blood banking next semester, one of the things that you'll be looking for is rouleau, and one of the things that you can use to disperse rouleau is using saline or mixing saline in with your red blood cells. But you'll learn more about that when you get to blood banking. On our last page, page 106, that we're going to be covering today in our atlas, it's talking about autoagglutination, or a lot of times we just refer to it as agglutination. And you'll be using this a lot in blood banking as well next semester, is seeing if red blood cells are clumping or sticking together as far as your results in your testing. But both of these images, figures C and D, is showing red blood cells just sticking or clumping together, and that's called autoagglutination or just agglutination. And again, you can disperse agglutination with saline as well. I hope you've learned a lot about the different shapes and sizes of red blood cells. I strongly encourage you to look back through your atlas and make sure you know the difference between the different shapes and the different sizes of red blood cells, especially know the difference between anisocytosis and porculocytosis, and we'll talk about it some more in lab.